Hello everyone, this is me Additi and this is my colleague Tushar, and today we are here with Dr. Rattan Deep Singh, who is a professor at Lovely Professional University teaching in the Department of Molecular Biology and Genetics. And today we are here discussing about some immense topic about aneuploidy and polyploidy with its application and with its type.
So sir, how you will define aneuploidy and what are its significance in genetics?
Well, aneuploidy, we can say any chromosome number apart from the normal. So generally it can be increase or decrease in number. So there are so many types. If I will talk about decrease in chromosome set other than the normal, we can have here monosomic condition, we can have here polysomic condition. As far as significance is concerned, like we can get new traits. These polyploidy, monosomic condition, trisomic condition in case of plants, generally we study in case of plant breeding. So we get more and more hybrid varieties as well as improved varieties that can be disease resistant, stress resistance and so on. So that's why we go with this.
Told about plant breeding. So what's the mindset in this in plant breeding?
Yeah, well, there are various applications of plant breeding today. Traditionally, like we are creating hybrid varieties, genetically improved varieties. We are creating Aneuploidy, we are creating octoploid, wheat is a hexaploid, strawberry is octoploid. So these different ploidies, we are creating variations in this. The seeds are being particularly created, variations are being induced in the seeds. We plant the seed in the soil to get a new plant that will have some improved characteristics. So plant breeding has emerged as a very prominent field today to make some desirable changes in the crop. Various crops you will find, like there are so many varieties of basmati rice in the market. Many varieties, more than 20 to 30 different varieties. So how it basically came? This all because of the improved characteristics. You have some rice varieties that are golden in color, yellow in color, some long grain, short grain. So they have been created with the help of plant breeding technique so as to get some improved varieties.
So you have also talked about previously about monosomy and trisomy. Can you elaborate on that?
Yeah, see, if I will talk about the monosomy, it is decrease in chromosome number by 1. If normal chromosome set is 2n, 2n minus 1 will be monosomy. And in case of trisomy, definitely if you are saying about trisomy, it is 2n plus 1 will be. In trisomy, there are examples like Down syndrome. It is due to trisomy of 30th chromosome. We have Patel syndrome, Edwards syndrome with trisomy of 18th chromosome. And moreover, apart from these monosomy and trisomy, we have various other sex-related disorders as well, like we have Klinefelter syndrome. Okay, so they are just happening today. Like we are making some improved varieties. So these mutations or these different ploidies we have to go on.
So these mutations can actually cause severity in the physical form of a human being or of an organism. Can you please explain about that?
See, basically we are not inducing mutation in humans, we are inducing mutation in plants. Now, the basic thing is that mutation can be harmful as well as useful. And if we are inducing a particular mutation in the DNA of a particular species and we get a maybe improved variety, I can give you example. There are various like Arabidopsis thaliana is there. It is specially genetic engineering plant that we have created with respect to resistance of plant against drought conditions. So we are creating the stress resistant plant, drought resistant plants with the help of these breeding and genetic engineering techniques. So they have a significance.
Sir, you told. So there's one more term, polyploidy. What's that and how it's different from aneuploidy?
Say polyploidy, generally we are inducing it basically. So we use colchicine to induce the polyploidy in case of plants. It is basically endoreduplication of chromosome. So polyploidy gives us improved varieties with increase in chromosome complement set as compared to normal. Normal chromosome 2n diploid is there. We can have 4n tetraploid, we can have 6n hexaploid, 8n octaploid. This is a polyploidy in which we are increasing the chromosome number in the plant, particular species.
There are two types of polyploidy like autoploidy and alloploidy. What's that?
Autopolyploidy is basically increase in chromosome number or chromosome set of the same species. In case of aneuploidy, it is between the different species. So this is the difference.
So sir, can you please elaborate more?
See, basically if I will talk about autopolyploidy, Suppose I have taken two varieties of wheat, Triticum aestivum and Triticum monocoque. Okay. Triticum aestivum is 2N, Triticum monocoque is suppose 3N. So if I will hybridize these two varieties, I will get 5N. Now, these two varieties are of same species, not Triticum. That is autopolyploidy. Now consider I have taken another example. I am taking on one side Triticum, another side I will take maize. That is two different varieties, no? I have hybridized chromosome of two different varieties and resultant offspring have shown increasing chromosome number. That is allopoly.
So it's between two different species.
Exactly.
Okay.
For allopoly.
So how can you induce polyploidy in plants?
Well, polyploidy can be induced with the help of some chemicals like we can use colchicine. Okay? Yeah, colchicine, we can use acridine orange acid. But colchicine is widely used. What happens here, like you all know about the mitosis?
Yes.
Okay? So there is duplication of chromosome. Now, if we are using colchicine, it arrests the mitosis and there is endo-reduplication of the chromosome. As a result, division stops and then there is abnormal division from 2N to 4N. Okay? So this is being done with the help of colchicine. So this chemical is widely used for inducing polyploidy.
So basically in meiosis what we do,
Mitosis.
basically that is a
Cell division.
So what happens is that they multiply and they divide, which we have the equation.
Exactly what happens during metaphase stage of mitosis. You all know chromosomes are at the equator. Spindle fibers are attached with the chromosome centromere. They pull chromosome towards the poles in anaphase. Now, what this colchicine do? It basically interferes with formation and attachment of spindle fiber with the centromere. As a result, chromosomes are not able to go to poles. Anaphase stage is skipped. And in the metaphase stage, it duplicates. Duplication is already been done here, no? Now spindle fiber formation is not there, because colchicine we have used, it stops the spindle fibers. And as a result, anaphase stage will not be there. And obviously, when anaphase stage is not there, chromosome will not separate. It is duplicated there. This is the way polyploidy is induced.
Sir, what you said is something which sounds like GMO crops. How it differ from polyploidy?
Well, as I explained you just now, like polyploidy is increase in chromosome numbers at the case of a plant of the same species. Now, genetically modified organism is different. In this case, we are transferring a foreign gene into a host cell, and the host cell is genetically modified. In polyploidy, genetic material is same, DNA is same there, but here we are modifying the genetic material. So, genetic modified organism may have two types of DNA. One host DNA, his own DNA, or another foreign. So there is mixing of genes, integration of DNA into the gene. Whereas that polyploidy, genetic material is not altered. In genetic modified crops, we altered the genetic material. This is the difference.
So you have just explained us the difference. But what is the difference between a polyploidy plant and the genetically modified plant? Because at the end result we are getting the modified plant. So what is the actual difference?
It's basically like previously we are going with the plant breeding technique, polyploidy only. So we get some improved varieties, that's fine. Now in case of genetic modification, genetic modified crop, we are also getting improved varieties. But the characters and traits are different. I can give you an example. Suppose there is a cactus plant. We all know cactus grow in a desert. Cactus needs some high temperature. Cactus doesn't been grown in a plain. Or cactus we can't grow in the Himalaya regions. Why? Because conditions are not there. So what we can do? We can modify the genetic makeup of the cactus. I can transfer any cold resistant gene in the cactus. Or I can even transfer any particular gene that make the cactus resistant to cold and give the same condition that we have in the desert environment. That can only be possible with the help of genetic modifier, through genetic modification of genes. Because a foreign gene that I will insert into the cactus plant, that will express. But in case of polyploidy, we can make the desired traits, okay, but we cannot make any like stress resistant plants, or maybe any, you can say, any plant that is being cultivated in Himalayas, we grow in a desert region. or any plants that has been cultivated in desert region we grow in Himalayas or plain. So that cannot be achieved with the help of polyploidy. So that's why genetic modification is important.
So can we say like that in genetically modified plants, we are choosing the own characteristics that we need in a plant, in a new plant. But in polyploidy conditions, we cannot choose the characteristics that we need in the modified plant.
Yes, exactly. Because in polyploidy, if I will say we are going for duplication of chromosome. The chromosome number is duplicated, but we cannot say whether we will get the desired trait or not. The traits can be any in this case. No? But in case of genetic modified crop, we even in this case as well doesn't know whether our gene will give us express or not. But we know that suppose this particular plant is having a particular stress resistant gene, let us transfer that gene in another plant. Okay, so maybe there is possibility once that gene is transferred, it will code for a desired character in the second plant. Okay, and the gene is expressing and plant will be resistant to some particular disease or maybe anything. We are doing it now, like there are so many diseases that are being affected plant, red rot of sugarcane, Fusarium, oxysporium, wilt is there in the plant. So what we can do? We can modify the plants basically. Okay, modifying the plants with a particular gene may code for a protein. That protein can be a toxin for the pathogen. As a result, once the pathogen attacks the protein, it will get killed. So that can be achieved with the help of genetic modification only. That cannot be achieved with the help of polyploidy. Polyploidy, we can give some plant varieties that have more nutrition, that can maybe cultivated for shorter photo period or maybe any longer period. We may need some less fertilizer, less pesticides in order to cultivate the plant. That is being done through polyploidy. But if we have to express a particular desired character for a resistance against some disease, then we have to go to the genetic modification. This is the difference.
So sir, we will now end this interview by the one last question. So what do you think economically which is better?
Economically, polyploidy is like better. Economically, because it takes less cost is there. Although, but if I will talk about, we get to get improved variety, then genetic modification is better. In India, generally, if I will tell you, only BT cotton has been used for the genetically modified crop. We are not able to grow other genetic modified, but we are dependent on the plant breed. So I will say that polyploidy is more prominent in our country. Hybrid varieties we are getting more as compared to genetic modified. But in world level, in China, USA, they are working on the GM crops. Okay, so they are getting the genetic modified variety. In India, we have made attempts for BT brinjal. BT brinjal was being made in India. Okay, it was being launched in the market. But we saw that it produces some toxins in the population. As a result, there are allergic reactions to the people. So we stopped the production of BT. Similarly, in BT cotton, the workers that are handling BT cotton, they constantly wear gloves. They wear gloves, mask on the face, because BT cotton initially was found to have allergen. The workers exposed to BT cotton were found to contain allergies. So they are using proper gloves, face mask, precaution once they are cultivating the BT cotton.
So thank you.